Hi everyone, these are the students from Biotechnology department and we are here to perform a role play on the replication of DNA. Deoxyribonucleic acid, what they call DNA, is the most important, common and found in all of the cells in living beings, and they hold the code for RNA. And DNA here needs to replicate because the cells in our body replicate, they divide and they form daughter cells. Where do you get the DNA? DNA needs to replicate too. So here we are going to talk about what DNA contains and how it replicates. Where does this replication take place? In eukaryotic cells where there is nucleus. During before mitosis and meiosis, this replication takes place during the interphase, gap phase of the cells. And let's get started. It has like 4 nucleotides, and they are adenine, thymine, guanine, and cytosine. These 4 nucleotides join together by hydrogen bonds. DNA strands, they are anti-parallel, complementary to each other. So they run anti-parallel with 3' and 5' end, which is called the strand and the ligase strand. During DNA replication, the first enzyme comes in, that is helicase. Helicase is the unzipping enzyme. It goes around and breaks the hydrogen bonds between the whole strands of the DNA. Now comes in primase. This enzyme sets the primer to make the new strands of the DNA in the old strands. Now comes DNA polymerase, which will join them together using hydrogen bonds. This is how the new strands and the old strands together form a new daughter DNA. The enzyme primase brings in the RNA primer and sets it with one of the old nucleotides in the DNA. And then it brings in other nucleotides as well. And it forms Okazaki fragments. Why? Because never does DNA polymerase bind nucleotides from 3' to 5'. It always binds from 5' to 3'. So now exonuclease comes in and replaces the RNA with a new nucleotide. Primase comes in and sets the nucleotide. After this, DNA polymerase binds the old strand